We are PamGene, and we specialize in measuring kinase activity. In this clip, we explain how our technology helps to understand cellular signaling. Let's start at the beginning. Cells are the fundament of all living things. They are the drivers of growth, adaptation, and defense. Cells are continuously responding to external stimuli. Upon stimulation, a signal is passed through the cell membrane to the nucleus, and this leads to genomic and phenotypic changes. Kinases are key players in transducing these signals. Studying kinases is therefore of great importance to fully understand changes in cellular processes. Kinases are enzymes. They transfer a phosphate molecule from ATP to a specific phosphosite on a target protein. This is known as phosphorylation. Phosphorylation leads to changes in conformation and can make kinases more or less active. Activated kinases will start to phosphorylate their targets, leading to a cascade of signals, also known as a signaling pathway. There are two major types of protein kinases. One targets tyrosine, and the other serine threonine phosphosites. We developed an array with peptides mimicking the in vivo phosphosites. This allows to measure activity changes of kinases or pathways in a lysate. When a lysate is incubated, the active kinases in the sample start phosphorylating their targets on the array. The amount of phosphorylation is visualized with a generic fluorescently labeled antibody that binds to all phosphorylated targets. A common application is comparing treated and untreated cells to look at the differences in kinase activity. We interpret which kinases are responsible for phosphorylating the targets that are significantly different between the conditions. The functional and multiplex properties of our chip allow us to display cellular signaling, and connecting these kinase fingerprints to a clinical response could lead to the identification of a predictive biomarker. Some other interesting features. Our technology utilizes the full-length kinases that are present in the cell lysate. A single fluorescently labeled antibody is used to visualize all phosphorylated targets. Our technology is sensitive, capable of measuring from 0.5 microgram of protein input. The majority of the kinome is covered by our multiplex assays. As fellow scientists, we are curious how our platform can make the difference in your research.